2.5 years.
That's a lot.
Please see the attached video.
Yeah, I mean, I'm not that. Yeah.
I was gonna say, what was I gonna say? I don't remember what I was gonna say. Dang it. Oh, I was gonna say thank you for doing this presentation. Everyone, today's presentation has been sponsored by Raid Shadow Legends.
Raid Shadow. Actually, it's been brought to you by Skillshare. Learn about basic neuroscience principles with use the code JoeyIsBald to get first 2 months free and 15% off your yearly membership.
That would be the best.
Also, Raycon earbuds.
And Raycon earbuds.
Perfect.
Don't forget about NordVPN.
I use Proton VPN.
I was gonna say hello to Mr. Yearn. I think you're from the main campus, Toledo, it seems.
No.
No, I'm not.
Who are you?
Who are you?
I am
That's
the
Josh.
chair. I am the chair.
Hi.
I am the slightly aggressively. No, sorry. I am the aggressively mediocre chair of
I see that.
PRC. Wow.
It's okay. It's okay. Josh he's a special child.
Yeah.
That's fine by me. So Adam has to drive home to Cincinnati, but he is tired. He got 3 hours of sleep last night, so he keeps.
He lives in Cincinnati.
He lives in Cincinnati now. Yeah.
Well, you could chain him to a radiator. That way, it would solve all your problems.
What?
Just chain him to a radiator.
Kyle told me to chain you to a radiator.
Okay.
It's fun and it's free.
I mean, I don't usually do that, but you know.
I advocate for chaining people to radiators.
This is why we can't have nice things.
That's because you're chained to a radiator.
Well, okay.
You can stay and rest for however long you want. You should rest. You can just go ahead and get started whenever you want, Joey, at this point.
Well, awesome. Okay, so I started off by titling this why I think I started with electrical engineers make great neuroscientists, and the more I thought about it, I realized that computer science and engineering is actually so perfect for neuroscience and modeling it that it's kind of scary. And hopefully by the end of it, you'll be like, wow. So basically, it's just like the computer science and engineering of the brain. It's kind of ridiculous. But so a little bit about me. I am a researcher at Case Western Reserve University, and I've been studying electrical nerve block for the last 2.5 years so far. Even though I am an undergraduate, I still have a pretty sizable amount of research experience from that, including a poster, and I actually have a publication now. So I think I am allowed to call myself something close to. I could probably say I'm knowledgeable about the field. I wouldn't say I'm an expert, but I can say I'm knowledgeable on the field. And one of the things I noticed is when I kind of started my work, you know, I knew all the computer science, and I knew a lot of computer science and engineering stuff, but I didn't know a ton of neuroscience stuff. And the more I learned, the more I realized that computer science and engineering set you up really nicely to learn about neuroscience. And so I thought this might be something cool that you didn't realize you're probably pretty good at, even though you've never seen it. And it sounds like it's something completely foreign to you. So that's this. That's where I came up with this idea. Feel free to always interrupt me because I get boring and I don't like listening to myself too much. It works when I'm running lectures or labs, but it just gets boring after a while. So,
but you have such a chocolate silky voice.
Is that why you're so quiet in lab all the time, Kyle?
Yeah, I use you as ASMR.
Well, that's good. Okay, but okay. So what are I tried to, because you know it's a very obscure kind of topic. I wanted to make sure that there's an actual direction to this, and I'm not just spitting facts at you and hoping they stick like spaghetti against a wall. So the goals of this thing are, you're going to understand what a neuron is, the basics of it. and how the electrical signals go across the neurons. You might already know a little bit about this, but we'll kind of cement it in. Then from there, we'll look at how you can model this, and that's kind of where we'll start looking at circuitry. We're going to dive into some math, but I'm going to try not to scare you. Okay, I'm going to try and break it down, make it nice and simple. There isn't a test at the end, so don't worry. I'll try and show you the math without, beating you over the head with it, and then we'll look at how we can code it. And then maybe if we have a little bit at the end, we can talk about where this could be going in the future. So, the electronics of the brain. So this is what we're going to talk about tonight. This one cell here, which is called a neuron, has a couple different parts. It has dendrites that receive signals from other neurons, which then go through, they travel along the axon to send signals somewhere else.
It's like a node in a graph.
It is exactly like that. And you can think about these dendrites and the axons then branch out and connect to dendrites. So these are like the what are they called? The connections of the nodes in the graph.
Like the pointers or
Yep. And these are so it would become a directional graph, because at least most of the time when you think about it, the electrical signals really go in one direction. They receive here and they go out here. I think there's been studies to show that when you sleep, it can go the other direction, but I'm not very versed in that, so I'm not going to talk about that. For the sake of argument, everything in today is going to be, it's a unidirectional cell. So what are some things?
So we think backwards in our sleep.
That's what I've heard, and I don't know what the actual study went with it and how they measure that. But I remember seeing something very strange at one point that was like neurons fire backward. So I'm not going to stand by that with a lot of certainty, but I'll say it's something that I've read at some point, and you can look into it later.
Are axons polarized?
Yeah, so we're going to talk about that. But there's essentially what's happening is there's ionic concentrations inside and outside the cell, and they're different concentrations. And the movement of those ionic concentrations are ions moving. And if we think about, what is current is charge over time moving in a place. So that current is it's the ions moving in one direction, and that's because there's this ionic concentrations in various locations, and we'll get into that. But yeah, so they move across, and there's ions kind of all over the place in a bunch of different ways.
Can we use them to rectify AC signals?
We use them to rectify AC signals. So part of it, I guess that kind of goes to this next
like
one.
the diode bridge rectifier.
Or just a single bridge rectifier, it doesn't matter.
So, kind of. One thing to note is it is an all or nothing signal. And what does that mean? It means when we get a sufficient amount of signal in, we get a single pulse going out. Or maybe we get a bunch of signal in and we create a bunch of pulses going out. But there isn't really like a fine tuning of like, we send like a half size pulse or like a 3/4 size pulse out.
So it's like DLD, it's either on or off.
Yeah. That's really how we think about it. There's kind of it kind of builds up, so it's like charging a capacitor and then it'll discharge in one kind of swoop, and we'll look at that. That's a pretty easy way to think about it, at least in terms of the all or nothing idea.
Okay, so are they completely conductive, or is there a dielectric effect going on?
So they have a capacitance across the membranes, and that capacitance across the membranes also fields into this all or nothing portion of it. There's voltage gated channels that we'll look at that allow certain times for ions to go in and out and things. and it's kind of cool when we break it down into ions, which is what we'll do in a few slides. So, but those signals will travel along, and then they go to what would be called a synapse. If we had kind of a connection from one end to the other of one end of a neuron to the beginning of the next, we'd call that a synapse. And that's how they're sent from one neuron to another. And then the neurons kind of, if we have like 50, what is it? There's 10 trillion synapses in the brain. So there's a ton of connections that are coming together to different portions that are coming together to different portions, and they're always firing, creating, quote, thoughts, or however you want to do it, or call it at least. And one other weird thing, which you might know, but you haven't really thought about, is that neurons aren't just for humans, it's all intelligent life. The same cell that makes humans smart is the same thing that makes apes smart, is the same thing that makes the slugs smart, or worms smart, or a fly smart.
Oh, how many terabytes would it take to store the state of all of the synapses?
I feel like there's a number that I'm sure somebody's come up with a number, and I'm assuming it's I want to hope that it's on the exabyte scale and not the terabyte scale.
Wow.
Interestingly, smaller organisms like C. elegans, which if I remember correctly, C. elegans. Yes,
I
the
just Googled
roundworm.
it.
Yes.
It says that it's so the average adult human brain has the ability to store the equivalent of 2.5 million gigabytes of digital memory.
So 2.5 exabytes.
Yeah.
Sweet. Wow, that was a pretty good guess. Okay. The other thing, so there are smaller organisms, one of them being do you guys see the wiki page I pulled up?
Yes.
Awesome. So one of them is called C. elegans. And if I remember correctly, one of the cool things we've done about them, if it's not this, there's another type of, I think it's a sea slug, but we've mapped out its entire, brain essentially. We mapped out all of the connections in it, and then we can model it in its entirety, which is kind of interesting to think about. I'm not sure if it was this one or maybe it was some other one. But yeah, so there's smaller organisms that we have started working on that. And one of the things that one of my particular interests is working on machine learning algorithms that are going to be closer to this type of structure and calculation method. So, but yeah.
So does sheer number of neurons separate like humans from apes, for example, like their intelligence? Like apes can figure problems out just like humans can. Is that related to the number of neurons?
So, I'm not entirely sure if it's purely based on number. I think that there's a lot of factors that go into play, and I'm not entirely sure if it's just number. I can't remember, like, part of me wants to think that, like, whales have a lot of neurons, but I'm just speculating at that point. Um, I'm not entirely sure off the top of my head why if it's a particular it's number, or if it's the connections. We have a lot more connections or something like that.
Okay.
I would also assume that structure plays a pretty strong role too, because I mean, like a dog can process smells way better than a human can, and their brain is structured in a way to be able to handle that. And people can process like abstract concepts more than a dog can. So I would assume that, I'm just guessing, but I'd assume there's like some kind of structural element to it too, not just like number of neurons.
Yeah. Yeah, I would completely agree with that. But yeah, it's interesting to think that it's the same. If you want to think about it as, before this, we were talking a little bit about transistors and things like that. There's kind of one component that like makes all the other things. This is kind of, I call it the building block. You could also call it, it's like the transistor, it's doing some logic. It's a different way of doing logic, but it's still kind of a logic piece. So, um, why don't we jump into a little bit? We talked a little bit about the ions moving across things. I'm going to break this down, so don't worry if it's like filled with a bunch of stuff and you're like, what the heck's happening? So, what I'm going to walk through is we have ions, some of them go in, some of them come out, and then they kind of get balanced back, and that causes what we call an action potential. So that's what you need to get from this slide. Ions go in, ions go out, they go back to their resting state, and then we can do it all over again. And so we start off with what's actually happening here is if you see there's like a plus across this side and a minus across this side. We have mainly a lot of sodium on the outside and a lot of potassium on the inside. Both of them are ions and they're positively charged. There's also other ions like chloride ions and magnesium ions are there. Chloride gets a little annoying because it's on the opposite side of the periodic table, so it's like a negative charge instead of these are positive charges. But what we start off with is there's a weird, kind of phenomena that happens where obviously we have a high concentration of potassium here and a high concentration of sodium here. So the ions from the concentration want to go one way, but the electric fields that are created by that push them in another direction. And we come to a kind of a resting potential where the ionic concentrations are not equal, but also the electric fields aren't equal. And there's a lot of math you can get into for calculating like what's the resting state of this particular setup where you have, one concentration on the outside, one concentration on the inside, and it creates a potential across it which, electric fields would want to move. So it's it's the differing concentrations that create what's kind of weird is the stable resting state of this is then at a negative potential, a negative, 70 millivolts, um, which is interesting to think about. So it stays at, negative 70 millivolts pretty consistently. But what are all these extra things we have here? So let's go through it. So let's say we start giving it some charge. One synapse starts feeding in current, so into the cell. So now the voltage inside the cell starts going up. And what happens is when it starts getting positive, we have voltage gated ion channels. What does that mean? At some voltage, the ion channel opens, but it only lets particular ions through. In this case, the sodium ions that were on the outside. When we start raising the potential in here, the sodium ion channel opens, which allows us to have sodium ions rush in. And if we think about that, we now all of the positive charges that have rushed in, which means we went from being a negative, usually around 70 millivolts. From negative 70, we go up to a positive 40 millivolt. Now, these are kind of, they're, there's not an exact number for any particular value, but these are kind of like the, average numbers that we think about for these voltage values. But that's
so
what
it's
happens.
actually letting charged ions through the gate rather than
Yes.
like just like pure current and voltages like in a computer system.
Yeah. Instead of thinking about like, we usually have electrons that move in places, and it always gets annoying because like the electrons flow in the opposite direction of the current, and we have to always go and think about that. This one's kind of nice because they're positive charges that
Yeah,
just
it's
go
still
in.
like moving electrons around, just like having them stuck to a sodium ion or something, like having them attached to a larger particle.
Yeah, except we have to remember that sodium and potassium ions are positively charged.
Oh, right.
Yeah.
I hadn't
So
thought
this is
about that.
it's a positive
Oh,
ion
so
moving
it's
in the direction
actually
of
like
light.
conventional current is actually the
Yeah.
way the particle. Oh, that's cool.
Yeah. Which is it's nice. Don't worry. They mess up they annoy us with other parts. We actually don't think about this as resistance. We'll think about it as conductance. So it will don't worry. We'll annoy the electronic people in the room later. But for now it's nice. We get to think about the positive ions is the current of is the actual current, which is nice. But so in this, the positive ions have flowed in, and so that's the sodium. Now we have positive ions flow out, which is the potassium. And so now you'll notice, oh, well, now we have the potassium rush out, and the potassium is then going to cause us to actually go back to the negative 70 millivolts because we have positive ions leaving. So now we're going to drop in potential, and we'll actually undershoot that negative 70 millivolt resting potential. So we went from negative 70, sodium rushes in and we shoot up to positive 40. Then the potassium rushes out and we'll drop to negative 80. And then we use the sodium potassium pump to send the sodium back out to where it's supposed to be and the potassium back in. And this causes us to go back to that resting state. And so if we go to this, you might be able to see this. We start off at that negative 70. The sodium rushes in and we shoot up to a positive 40. The potassium goes out, and so it drops down and we undershoot. The other thing is when we undershoot, or here, and potassium's on the outside, sodium's on the inside. So we can't fire again right here. We have to wait. We have to wait for it to get pumped back using the sodium potassium pump back to our resting state. So this what we call the refractory period, we've fired an action potential and we have to wait before we can fire again.
So it's kind of like discharging a bouncy chemical capacitor.
Yes. It's kind of it's neat that it does that. Um and you can kind of force it to fire faster sometimes, but there is like a limit to how much you can do it, which is part of the interesting thoughts of why is this an excellent way to uh, work with data in our brain. Why is this the way that has come out as why we fire things, as why we think of things. um, this is the way intelligent life does its stuff, and that's really it's kind of magical. Really, it's magical. But also, I thought it's also cool to watch an animation of it, so I made a nice animation. It made me very happy. But if you watch, this is an action potential. So this is the entire length of the axon. So if you remember, it's a longitudinal thing, and the at each point, the sodium rushes in, and then when it's starting to go down, the potassium is rushing out here because we're dropping back down. But
So,
when sodium rushes in, it causes the next one to have sodium rush in.
so is the axon just like a long string of synapses?
So the axon is a long string of the channels. The synapses would be at the ends. So they would get
Got
the
it.
input or send the output. Um, each node is where the channels are, which is where the ions flow in and out. And the depolarization of one node, remember, there's like a capacitance across it. And there's also a resistance along the axon, which causes, if one node increases in potential, the other nodes around it, because it's like a circuit, are also going to have some amount of increase. It'll be slightly less because, there's a resistance along the axon. But if you think about it that way, there's still this increase in potential, which causes the sodium to rush in, which causes a huge increase in potential, which then travels along the axon. And you get this very nice, very kind of hypnotizing graph almost. So that's it. That's action potentials. This is how your brain sends information. Always has, always will. Until you die, and then it stops. But well, it's more fun. More fun.
How does it? I don't like. It's not like bits like a computer.
In some ways,
how does vision get to my brain?
So when you kind of have neurons that are at the back of your brain that are photosensitive or whatever, light sensitive, however you want to call it. And when those get particular amounts of light, they fire. And then all of those signals then get sent to your brain, all the way around the back, which I don't know, it's kind of a dumb design to enter it in from the back, but that's just me. And then they come in and they get accumulated across a ton of different neurons. And it is kind of like sending a bit because it's either not firing or it's firing. If you notice, like the axon, it shoots all the way up. It really doesn't shoot halfway up. I'm kind of saying that it does sometimes, kind of, but it's more of it kind of shoots all the way up or it doesn't. And then there's a synaptic connection, which has more information there because maybe it's like a partial connection, maybe it's a really strong connection. And that's where you can that's where really a lot of the information comes from, is these synaptic connections, because those aren't a binary type thing. Those are a much more analog connection.
So once this information is sent to the brain, how does it get stored?
So the way it can get stored is those synaptic connections. So there's a saying that goes along the lines of neurons that fire together or wire together. As neurons that happen to fire, and let's say, this is, one, if we're thinking about it as an image, sent to your brain, it's a pixel. That's not exactly how it would work, but for sake of an argument, it's a pixel, and then that gets accumulated with a bunch of other information. And let's say that leads to some neurons firing that, tell you, oh, you're looking at a cat, and that's right. And so because that led you to like a right answer, and those all fired together, the connections between all of those get a little stronger. And so the idea of, oh, you see all these pixels together, it tells you that's a cat, just became a stronger connection.
Oh, so you just like form associations between things.
Yep.
That sounds like a machine learning algorithm.
It's similar. What's different is a lot of machine learning algorithms, we force it to go, like we force the weight of a connection to change in a way that we know is going to be good. In our human brains, it doesn't really, it's not, at least as far as I've seen from the literature, it's not exceptionally obvious that just using the concept of fire together, wire together leads to the final, always correct brain connection setup.
Okay.
So I think there's more at play. I'm kind of interested in looking at what's the effect of feel-good chemicals in the brain in terms of learning? Because a lot of the feel-good
and serotonin and all that,
those cause your brain to either fire more or fire less, which affects the concept of fire together, wire together. And so can you combine those ideas with the chemicals in your brain and model that in a machine learning algorithm is kind of where I'm hoping to look into.
So like if I have a bunch of dopamine, if I'm in a good mood, then it makes sense that my neurons are going to be firing more, which means I might be more creative, more aware, more on top of things.
Yeah, I can't remember exactly what the particular chemicals do off the top of my head, but they affect how your brain fires, and that affects your thought process in general. So, do you guys like this slide? This is a good slide.
This is a baby squid.
So this At least it should be. I'm not entire. I actually don't know exactly what a giant squid looks like. It's actually not that giant. Um, this is where we get to talk about circuits. Is because of this animal. And you might be thinking, why? It's partly because they have really big axons. I think they go up to like 1 millimeter or something, which in terms of axons is pretty big.
Yeah, that's huge.
So that made them really nice to measure the currents across them and concentrations and stuff like that. And so, two people, Hodgkin and Huxley, um, worked together on their. This is really the, this is a revolutionary paper. It's a very important paper in computational neuroscience because they created a circuit that models that current going across that membrane, and that current being the ions that go across. And so we have the sodium current and we have the potassium current. So if you remember, the sodium current's going to rush in, which, if you notice, this is a positive battery right here and a variable resistor. So we turn on the variable resistor and we'll end up with a positive inside and a negative outside. The potassium's the opposite. If you remember, the potassium will shoot out. So we have the positive on this side, the negative on this side, and a variable resistor. We have a capacitance across it. And then we kind of just assume that there's other ions at play because we don't know everything. So this is kind of our, I wouldn't say it's a fudge factor, but this helps us maintain that resting potential, essentially. And this is it. This is the circuit for, well, this one's particularly for the membrane across a squid axon, a giant squid axon. But this circuitry is how we've looked at pretty much all neurons.
That's pretty cool.
So you just have a capacitor and then some variable resistors, and it's a circuit. Look at that. You guys already, you know what it is. And if you needed to, you could probably calculate things if I told you what the resistances were and stuff. So yeah, you guys basically know it. This is the end of the present. No. So, well, this is the axon. So this would be the axon. Then you'd also have to make a connector thing that goes from one axon to another to make the synapses. But yeah.
And I guess a way for them to change their connections based on past experience.
Yes. And if only we had a nice new piece of technology known as a memristor, which allows for a variable resistance for the synaptic connections that we could change, and that would allow current to travel through at some variable amount. So now you know why I'm interested in memristors as well. They form the synapses that would happen here.
Do they exist?
Yes.
Weren't you offered a position as a graduate student?
That is where I will be in 6 months.
Oh, you accepted that one?
Yes.
That's crazy.
So I'm excited.
Can you take me with you?
No, I'm going to be living on a graduate student stipend. So
yeah, me and my son can come and live with you.
If you want to eat ramen all day, then maybe.
We like ramen.
I know.
It strengthens the bones.
Too. Yeah.
That's what they keep.
I call it top bunk.
Okay.
I call middle bunk.
Okay, it's time for math. Are you guys excited? I'm going to break this down. This entire slide is going to be filled with math at some point. So
Josh
you need to be mentally prepared for that. Yeah, I know. So the current across the membrane is equal to the 4 components. We had the current that's caused by the path through the capacitor, the potassium channel, which we mark as a conductance times the voltage. And we'll talk a little bit about what this n to the 4th is and what this m cubed h is. We have the current through the capacitor. We have the current through that potassium channel. we have the current through the sodium channel, and then we have the leak current. And each of these currents is broken up as a maximum conductance times the voltage across, which makes sense. The voltage across the 2 points, including the battery, right? We have the battery. The current through this is the voltage across here to here minus the battery's voltage. And then we have some variable conductance. And that variable conductance is defined as a maximum conductance times the amount the channel is open, which we use with these n to the fourth is a value from 0 to 1, and this m cubed h is a value from 0 to 1. And so that's how we do it. So these gates are going to be opening and closing, which change the amount of current that we let through based on the voltage across and the where the ions want to go. And so that's that. So we break down all of those gating variables. So each gating variable, there's one for the potassium. The potassium's a slower channel. And so it kind of opens in one kind of way, and then it slowly closes. So we use an n to the fourth. The math is where it gets complicated. So, and then we have an m cubed h, which are both for the sodium channel. M and h are two different gates. The m is the opening of the gate, and then the h is used to close the gate. And we come up with these great equations. We have alpha of n is based on the voltage and some other stuff. And if you'll notice, so these are all differential equations that are based on the voltage. But if you look at this equation, the voltage is then also kind of based on the gating variables. So it gets into this excellent mess of equations where we come to this. So this is all of the equations for all of those alpha and beta equations. So this is all of it. We have the currents for each of the channels. Each of the sodium and potassium channels have gating variables that are defined by these equations, which are further broken down into these particular equations. It's simple. Right? Yeah, it's simple. Understanding the math isn't important. Just knowing that it's basically a circuit and you could figure out the math if you needed to. Really, you never have to figure out these equations. You just put them in, your whatever MATLAB program or whatever we're using to write this, to simulate it. Somebody else has usually written the equations. Occasionally, people kind of come up with their own models, but they pretty much always stick to this format of we set up all the channels, we give them gating variables, and they pretty much always set them up in this format of alpha of some of the voltage times 1 minus the gating variable minus the beta, and then they change kind of these portions of the equations like the 0.01, or they'll change the 0.125. They'll adjust those values to whatever particular experimental data they're trying to match with it. So the whole point of all of this is this set of equations is supposed to match what was experimentally found with the squid axons. And this was back before they had so this was 1950. I want to say 52 or 58, and I'm not sure why I'm forgetting which year it was. But that was kind of before you could just easily run a set of parameters across your laptop. So try imagining doing all of this without a laptop in front of you to run the math.
I'm imagining doing it all on punch cards.
I can't even remember how they did it themselves. It's kind of crazy to think that they were able to do it all.
Hope you brought your slide rule.
Yeah. And it's a bunch of differential equations. So have fun doing that one. Yeah, 2 slides. That should be more than enough computing power. But the point is not that you memorize the equations. It's more that you understand how they fit together. It's a circuit. we sum it up like a circuit, we break the components down into equations that make them match experimental data. And that's it. That's the gist. So the other thing is this isn't the only model. I kind of talked about and alluded to the idea that, when they get input, it kind of sums it up. We use this is a simplified model of integrate and fire. So it gets input, and it's literally just a downward trend. So it's like a capacitor discharging over time. And as the capacitor gets more input, it charges up. When it reaches some threshold, we say it spikes, and we reset the capacitor. And so that's a very easy model to, we could wire it up and have it do that, and that'd be pretty neato. Then there's also more complicated models. So this is the one that I use for a lot of work, at least in my particular field. And it's based off of human, cat, and rat data. And it includes a bit more circuitry. So now we have 2 sodium channels. We also have different compartmental geometries. So we have, nodes with all the channels. Then we also include that myelin sheath that's on certain types of neurons. And that has different properties. If you notice, under the myelin, there's no channels. It's just, there's just that resting potential and a capacitance, and then it's a similar concept with the myelin around it. But we won't get into that too much. Just know that there's a lot of models. Some of them are much simpler. Some of them are much more complicated. So let's code. And by code, I mean do a little bit more math. This is the last slide about math. And if you remember, we had a bunch of differential equations. How do we simulate differential equations? Well, they're too complex for us to solve. There has been work where they make simplified models off of those equations. They make simplifications. And then with that, they solve it analytically. And you can do interesting things with that. But in the complex models, you can't solve them analytically. So we're left by doing approximations. And usually you're using, forward Euler. Actually, typically you use backward Euler or Crank-Nicolson. If I remember, Crank-Nicolson allows for a variable time step as you go through your thing. But basically, we look at the slope of any one point to determine where the next point approximately is. And then we take the slope of that point to determine where the next point approximately is. And so obviously, we have to take into account the rate at which a neuron can fire. How fast can that voltage go up and down? Because our approximations of it have to be, relatively close to what we think it is. So there's you have to know a little bit about how fast things move to make sure, one, you're using the right equation, that's part of it. You're using the right time step to make sure you're approximating it correctly. But that is all of the math. We took all those crazy differential equations, we used this fancy math stuff, and then we could actually code it up. And there's a couple different options. You could go hardcore and code it in C or C++. I actually don't see this one very often, mainly because it's a lot of work. And usually you're kind of testing one relatively small thing, so you don't necessarily want to do a huge program for it. You could code it in MATLAB or Octave if you prefer open source. But MATLAB, obviously has a lot of them who aren't necessarily programmers, because if you think about it, a lot of these people are neuroscientists. They usually came from a more biology-esque background. They're not hardcore programmers, so they're probably not going to do C or C++ to begin with. And MATLAB has a bit more of it's more of a calculator feel as opposed to a full programmer object-oriented feel. It's also computationally efficient for math and matrix math, which you can break up these differential equations into matrices and do it that way. Or you could use the neuron simulation environment. There's a whole book written on the software, which I have had to read. It's a riveting tale, a lot of twists and turns. and you can use Python. You could write theoretically your whole thing in Python, but I use Python to interact with the neuron environment, which does all the simulation stuff for me. It takes the time step math and does that for me, and things like that. So this is how you'd code it. You'd pick one of these, you'd write up your equations, and you hit run, and then it would evaluate them over time. And that's it. Here are some further links. My code for work. This is a link to this actually has some sample code for MATLAB, and I think maybe some other languages to simulate neurons. I have a video playlist for using my stuff, and there's actually a model repository that a lot of researchers, when they make a new model, they can post it there so that everybody else can see. So that's everything. So I guess going back to oh wow, there was a lot. Okay. Going back to the beginning, hopefully we know what a neuron is. It's the thing that sends information. That's it. That's everything. Any questions? You guys ready for the test?
So is this how YouTube knows what video I'm going to watch next?
No. So this is one of the things that I kind of don't like about machine learning is that if we look at a neural network and I look at the images for it, we have this standard thing. Now the problem is that this is kind of the interesting way of how this works is we set up the neuron, or I'm going to call them nodes, where they have input and then the synaptic connection, we have some weight, and then they get summed up together. But if you think about it, they get summed up, but it's not an all or nothing type thing. It can be a partial connection that goes in. And
so it's different.
So it's different because it's not an all or nothing. It's not doing that kind of strangeness of it doesn't seem like it should be as effective. And so because we do it with that, and then we also use mathematical things to force it to go into the optimum way. You can think about this as a group of neurons, and then that has a weight to another group, and that could be defined as how you get that partial amount of firing. But I think that there's something to be said about making networks that are specifically on the individual neuron scale, and instead of using complex math principles to get there, how does the simple biology get to the correct answer? The other thing is that we use megawatts of power to calculate these machine learning algorithms and train them and have them run, and your brain runs on like 10 watts. So 10 megawatts that does amazing image recognition, video processing, voice recognition, complex calculations, and abstract thought for 10 watts. And then we have supercomputers that have a hard time telling the difference between a picture of a dog and a picture of a dog and a cat in a picture and figuring that out. And those are using 10 megawatts of power. And maybe there's, a difference in paradigm that's kind of going on with those two architectures.
So what are some interesting things that have been done with like coding based on actual functioning of neurons?
So I guess I could talk about my particular work. One of the things we do, this is actually the paper that I'm on. So we can look at this as an axon. This one's a little hard to see, but what I'm doing is, do I have a nice sample? So we have this is the experimental setup. We have a rat. We put a blocking stimulation on the nerve. So this stimulation is such that it stops action potentials from going across. And then we have a stimulator here that sends action potentials all the way to the muscle. And so when we block them, the muscle doesn't contract. When we don't block them, the muscle does contract. So if we go to, is there a nice blocking image? So if you go like here, if we sent signal, we have test pulses here. If we sent them during this, we wouldn't see the muscle contract. But one of the things that we can look at is we can actually model that in the circuit. I hate when it does this.
So it's actually like a neurological transplant.
Kind of. So here's an example of the actual. So you can see that test pulses are these arrows here, and they don't create action potentials. But if we look at here's the modeling.
Or prosthetic, I mean, not transplant.
Yeah. Sometimes, or they could be implanted, or I think we're working on like surface electrodes. But so on one end, we fire an action potential. So this is the test one, and we can simulate that blocking stimulation, which blocks the action potential from traversing the other side. So it's only like a little blip instead of the actual firing. And so we can look at that in the model. And the cool thing about the model is that we can look at what's the sodium current and what's the potassium current, which are really hard to do in the rats because those have much smaller axons. And with that, we can start to conjecture, why does this work? We can look at the mechanisms for why this works, which you can't really look at on the rat without doing a bunch of more complicated things. Whereas I can look at it and say, oh, well, when we do this particular thing, the sodium channels are in such a state that they can't continue firing. Or you could say, we model it and we keep track of where the ions are, and we can say, oh, well, when it's in this blocking state, all the potassium is getting stuck on the outside and it can't fire anymore because, there's so much potassium it depolarizes the cell. And so we can look at the mechanisms to understand what the actual in vivo experiments are, what's going on within them. So that's kind of what the modeling is useful for.
So stuff like more closely related to actual neuroscience than to machine learning.
Yeah. Partly because it's a computationally expensive model, right? If I pull up the circuit, all of these equations are pretty complicated. And look at all these like exponents and stuff.
That'd really throw a CPU through a loop.
Yeah. And so if we had something hardwired, I think I've seen circuits that try and recreate these kind of voltage gated ion channel-esque properties. And if you had a piece of hardware that does it, then that'd be great. But to do all these computations in a traditional von Neumann architecture of running instructions on data and processing it, this would be extremely computationally inefficient for what we actually are trying to simulate.
Yeah, clog things up pretty fast.
Yep. Yeah. I mean, I run. Actually, is it still? It's still running. Okay. Oh, never mind. It stopped. So this took 3,805 seconds to run 1,000 milliseconds. So 1 second of this simulation took, what is that, 50 minutes for 1 second.
Wow.
And this is a single axon.
Holy crap. Yeah.
So there's kind of a problem with our architecture differences.
Yeah, that's not going to be predicting any song choices anytime soon.
Not yet anyways.
How did you get interested in this stuff? And what made you like find? How did you find the research at Case that you're doing?
So, so my dad kind of he, yeah, I'd say, yeah, he started the nerve block group at Case. And he's since kind of more moved into a. He's kind of like the guy that just comes to the meetings and gives ideas. um, but you know, he was the guy that wrote the initial grant for this back in 2002, 2004, or maybe it was 2001. But uh, yeah, it's been it's a good group. We do a lot of good research, but so when I was working at Highland, you know, he was like, "Oh, you should come and do some coding for me." And so I started playing around with Neuron, and you know, by the end of, you know, the semester, it was either I could choose to go back to Highland and keep being a software developer, which I don't know, was kind of boring for me. It works for other people. It just didn't get me excited anymore. And so, you know, I went and interviewed with my now boss, Dr. Badra, who was on the first kind of nerve block paper our group did with my dad. And so he was like, okay, well, you got coding skills. And, you know, I'd spent a decent portion of the semester while I was working at Highland trying to figure out how to use Neuron. And there are very few people that know how to use Neuron. It's not a large community. And so because of that, and I had, you know, I have good skills with programming and I know all the circuitry. And so that actually made me pretty good at, even though I didn't necessarily know all the biology, I could see how it translated uh, to the circuits.
What's been your favorite part about working there?
I think the best part about, I mean, it's more, I'm not particularly super drawn to, you know, electrical nerve blocks, cool, but I'm more interested in like machine learning stuff, and that's just me. So I've really enjoyed just the academic life and the fact that all I do is try and look for problems and questions to look for. And so I don't have a backlog of this is exactly what I need to do. I have my boss come in and say, "What would happen if you did something like this?" And then I respond with, "Well, I could do that, or I could do it in a slightly different way." And then accumulating data and trying to understand new things that people haven't looked at and just always asking questions is the part that I enjoy the most.
So really, nobody but your boss knows what you do, like you always said.
He doesn't really know what I do.
See, yeah.
um, I mean, he doesn't really write. He hasn't written code for Neuron in, you know, I think 10 years probably.
Mm.
And he's a he used to be an orthopedic surgeon, I think, in England. So, um, you know, he's very heavy on the biology side, and there's not a ton of people who really do this computational stuff. Partly because the computational work really needs to be based in the biological work, and so there are some groups that just do modeling, and it's kind of, you know, it just feels like it's missing something because it doesn't have the biology that goes with it. A lot of the times it should be there's a biological experiment and then we model it to understand what's going on. If you just do the modeling, it's kind of weird. And if you just do the experiment, you're kind of missing out on an understanding you might be able to get. And so that's exactly what the paper that I'm on is have a biological experiment that I then model. And we look at, oh, we can look at the data and say something particular is happening. And that's actually the whole direction of this paper. It's a particular phenomena we see. We see it in the animal, we recreate it in the model, and then we go from there.
How many people in the group are actually able to do the programming like you do?
Oh, I'm the only person that does this programming. I taught one of the other ones where they could start if they ever were needed to. But I'm the only one on my group that does it.
Who did it before you?
There was one guy. Uh, a while back, 2010, I think, 2007. Dr. Michael Ackerman, and he did some modeling. He actually did some interesting stuff. But when I got my code base, it was a lot of relatively semi-documented code with they're not programmers by trade, so they don't necessarily have the best indentation structure or naming conventions for things or coding structure. And so one of the nice things about being a programmer is that I was able to create well documented code that's a nicely presented style that I can feel good about. But yeah, there really hasn't been that much modeling on my group. I really do all of it, and part of that means I get to have a lot of freedom with it, and that's part of in academia. There's other people that do things similar to you, but rarely do you ever have somebody else with you that's doing exactly what you do. Even in our group, some people in our group are more interested in surface electrodes. Some of them are interested in the electrochemistry of the electrodes. So, that's a whole different part I didn't even talk about because it's not my forte. Where's the picture? If I scroll up to this one, we have to use electrodes that connect to it. Well, what do we make the electrodes out of? Because we fabricate the electrodes, because you can't just buy them off the shelf. Well, you can buy certain kinds of electrodes, but not these ones. And so some people are interested, how's the electrochemistry affect the block? And others are looking at, okay, how can we optimize this to do other things like that? Can we optimize the electrical signal going in to optimize the block? Can we play around with what other parameters can we play around with? There's a lot going on, and everybody can kind of pick a different part, and we all work together because, I kind of need to know a little bit about the electrochemistry when I'm matching the experimental data, but they don't necessarily need to know everything about the modeling, and everybody kind of has their own strengths, but you have to work together.
Sounds like they're going to be hurting a little bit once you leave for
I mean,
grad
you
school.
don't necessarily need the modeling, and it's available if you need it. So it's a nice to have, but it's definitely not the need to have.
Oh, okay.
There's not necessarily any portion of. I mean, technically, you kind of just, in terms of what you really need, is the experimental data is really needed. Whether you get that from a different group and you match their, so like we theoretically could have written one paper on our experiment, and somebody else could have done a modeling of our experiment, but that kind of gets a little annoying to do, just because then you're just doing all your work based on exactly what someone else does, which isn't usually what you try and do. But yeah.
Well, cool.
So yeah, so that's what I've been doing for the last 2.5 years while we've been in classes.
And you've been doing it constantly, right? You're always working part-time.
Yeah, I have been doing this every semester.
Nice.
Part-time. So.
Well, congratulations on 2 years of this. Thank you for the presentation. That was really cool.
Thanks for having me. Glad I could finally talk about it.
It's been a long time coming, actually, kind of, now that you think about
it. Yeah. It's kind of funny.
Well, cool. If there's no other questions,
what's your favorite soft drink?
Mountain Dew.
I don't know. I think I kind of like to mix it around a little bit. Like, I enjoy like the white monsters. I don't remember what flavor they're called, but I can't have those every day. I just feel like trash if I do that. So may mix it up with the new watermelon Mountain Dew. That's pretty good. May a Sprite. I'm not much of a cola fan. So, yeah.
What about Dr. Pepper?
Dr. Pepper. Yes.
Nah. I can drink it if I really need like a sugary drink, but I prefer like a fruity flavor. It's just always where I am.
So no Pepsi either, it sounds like.
Nope.
I can
Not
agree
usually.
with you on that one.
Yeah, I don't know what the difference is between Coke and Pepsi. They taste the same to me.
Pepsi tastes really good when your boss gives it to you at the end of a long work day in the heat. So
Yeah.
I
It
don't
has to
really
be
care
cold,
what-
otherwise it tastes like cat
That's
litter.
true.
Yeah, well,
Yeah,
it
yeah.
was very cold. It was good.
I'll buy it.
I liked them just because of that. I'm not sure if I would have liked them in any other circumstance.
Cool. Will the Mountain Dew hat ever make a reappearance, Joey?
The Mountain Dew hat, dude, it is like it's pretty old now, and I mean I still have it, I think,
I was about to
but I
ask.
realize it's like kind of big, and it makes my ears like flop to the side, and it kind of looks weird when I see it now. But I think it always did that. So
If we ever do another Halloween party, you gotta wear it and just come as freshman Joey.
a freshman. Oh, I'll have to shave the beard.
That's
No.
right. You didn't.
I'll
have
go
the
back to just sideburns.
I